Alrighty. So because of the modular, I get a gold star for using a buzzword. As far as the modular aspect of this orchestra, I don't have to be the only conductor. We can pass that baton around, metaphorically and physically, to others who can take on the role of being the leader of whatever type of sonic art we're creating. And so now we're going to bring up Courtney. And let's get up, set up for Cobra.
Scene and then the jazz scene. And basically what he did is he codified like a lot of different rules that musicians use for improvisation. And then he put them in like these cards that you had to follow. For example, I'll go through a few, and we're only using reasoning and reduce set. So here we have the pool card, and that means if you get this card, if you're a musician, you can jump in the pool or jump out of the pool or change what you're playing. The circle. And so here, this is the duo card, so that means I get to cue people to play with each other as a duo. And here we have a trades card. And so that means I can point to somebody and they have to start doing something, and then they pass it around the orchestra. And this is cartoon trades where you get to make a funny noise when I point to you. Okay, ready? Okay. Okay, it's looking good.
fun ones where I actually get to, participate.
One part where one of the guys asked me exactly what I was doing there.
Yeah.
He's like, so what exactly are you doing?
You can try hitting them from there
Just
too.
filming and like,
You can try
filming what?
You can try hitting them into the holder. Stop.
Expensive. What you can do here actually is the I don't know if you can't probably see it right now, but if you press up, you can see that line there, how it's starting to show up. These are referred to the pointers for the ball. The balls are like right here. You can't really see it right now, but what you want to do is actually move around the point there. The black one is going to correspond with the hole on the left, the white one with the hole on the right. And what you want to do is get your pointer in a general area of which way you want to go. And then what you'll do is actually swing the Wiimote, and while you're swinging, press the trigger on the bottom, and that's going to hit the ball. So line it up with what you want to do, swing, and then press. Yep. So there's the one, and I think that might be, that's yours? Okay. So yeah, you have enough of an angle where you can actually point it right at the hole, and it'll probably go in.
So what do I pass?
Let's see. I think yours is still right down here.
Ooh, is that you, Ryan?
Oh, there you go. Nice. 2 hits. That's actually really good. What you want to do is actually just continue to press up. That's going to change it. So you see how that works? So if you press it right, it's going to move it right. If you press up, it's going to go forward. And what you want to do is swing and then pull the trigger during your swing. Yeah, so Wii bowling. That's what everyone's been saying. It's pretty similar. But yeah, your hole is going to be on the left, so you want to
see where it is, Kira?
that way.
You gotta go in this one.
I think it's really funny because from what I've seen between the kids and the adults that are playing, the kids still get it, you know?
Oh, of course.
They pick it up easy, and I guess I haven't thought about it because I guess I'm not really old, but I'm not a kid either, and it's just kind of interesting to see the difference between everyone while they've been playing.
Do you see the little line that comes off of your ball, Kira?
Yeah.
Okay, well you need to aim it like you're playing pool or bowling into the hole. Here, Ryan will do it.
Yeah. So you want to have it right on where the hole's at.
Move it this way. There. Right there. Nope. There. Nope.
A little bit left.
There.
You go. That should be good right there. Okay. There we go.
Good.
All right. You guys want to try again?
Sure. You want to try again?
Kaden, you could go.
Let Kaden try. Here, I'll hold your back.
Wait, so
Hold
who
your
did
purse.
this? Who did this? Who made it go like this?
He did.
Hey, Jack. Do the kids have the golf balls? No. No, okay.
Holy cow.
They've been a bit
What is it?
Oh, it's supposed to be the 3-day festival.
Score!
Oh, they all got stuck inside. So now you get to use the helicopter in the car. They're pretty much
more to make.
Yep.
Because the merge that ASU that we do has all the.
You made
There's a
this?
festival. Yeah, we made it.
You made this?
Yeah, we made these.
You made this thing?
What the?
Right.
Did you just make it?
Yeah. All right, Kaden, Kira.
Yeah.
Kira, give me your bag. There you go. Good job, Ryan. Hole in one. Woo! Kira, it's called
Mini golf, not big golf. Mini golf.
Kira, little gently there.
Mini golf.
It's not baseball. That better.
Close. You got in there. You can try and get into this thing.
Hey, try to get over here.
If you get in here, it goes down the hole and out.
Kira, is that your own version of golf?
Yes.
She's doing a dance.
Yeah. Okay, Kaden, you want to try? You can try
If you
again.
try and
Kira?
get in the corner here, it'll fall down. And
Not so hard.
Oh!
When they push
Do you want to do a second?
really well or something. There.
Get stuck here.
Just get stuck.
One more whack into the hole.
All right.
Get your commemorative pin. Tee up.
You guys can see whether or not he's fit or not.
Can you see it? It has little legs on it. Can you see it? There are other things here for you to see also.
Molecule called PTC. And for some people you can taste it, and other people you can't taste it. It all determines on the types of alleles you have. You have a dominant allele, then you have a molecule on the good taste buds, and it binds better to PTC. If you have the recessive allele, then the molecule on your taste bud is conformed differently, and it doesn't bind to PTC as well. So if you take that piece of paper, you put it way in the back of your tongue, where your bitter taste bud receptors are. No. I mean, hold on to it. Don't swallow it. Yeah, put it back in your tongue. And some people taste bitter, and some people just taste like
You know, she can do it. She can do it. So I know.
No, it tastes like a weird taste.
weird taste. Maybe try it again.
There you go.
Taste nothing, or does it taste bitter?
Yeah, you don't taste anything.
Don't taste anything? Gotcha. There you go.
Right there.
It does taste bitter to you?
Tastes disgusting.
It's gross? Oh, you got a nasty one. Oh. Alright, go ahead. There's a trash can right there.
Why would you make me do that, you insane man?
Sorry, buddy. Alright, great. Well, thank you for joining on this part of the study. Now, have you been to the rest of the table? No.
And this is the coloring part, which is good. So first I want to see what my different size pieces of DNA are. What color they are. So my little pieces. What color are my little pieces of DNA? Okay, yes. Can you put that back into me? I gotta keep my DNA. It took me a long time to make it. Okay, so right next to number 1, can you put red? Right next to number 1 in this little box. Color red right in there. Good job. And you don't have to color the whole thing.
It's all like me, or it can be short, like someone who's short.
Okay, so now...
Our table is called It's a Small World. At our table, we are very, very, very excited about size. Okay, so when you hear the word size, does anything come to mind? Can you think of any other words that we use to describe the size of something? Length, feet, weight. So you kind of get the idea. So these are the words we use to describe size. Big, small. As scientists, we want to describe very, very precisely what is the size of something. If you wanted to know exactly the size of something, what might you do? Use a ruler. Great. So here I have 4 everyday objects. Paper clips, pieces of macaroni, grains of sugar, and molecules of water. On the second page of your workbook, I would like you to, I would like you to take a guess. How many do you think are in each jar? How many paper clips are in this jar? How many pieces of macaroni are in this jar? How many grains of sand or sugar are in this jar? And how many molecules of water are in this jar? If you guess the size of
You want to guess? Yeah, they go to
Do you have a bracelet? You want to go to the bracelet?
Bracelet?
What's your favorite color?
Yeah, it's just remember, you can get depends on
Also, pick one. You hold out your hand.
I think he's falling off.
Yeah.
Like this, we did like this.
Oh, are you getting all neoned up there, buddy?
Yeah. Oh, you can write?
What are you doing? Do you want to make your goggles?
Gotta have goggles.
You have to.
So one of the sides that doesn't have any shiny stuff on, it's better to mark on. Just to let you know, that's the hint.
Indeed.
Be able to detect the disease before initial symptoms. So the way we detect that is we've got your blood,
and then we apply your blood onto our slide. And if your blood has antibody is something that stick to different stuff. So if this antibody can recognize this protein, so that binds to it.
Oh, okay.
And then we'll use a secondary antibody, which will bind to this antibody. And this one has a fluorescent dye, so we can actually visualize what is binding on this spot. And if you have an antibody that is correlated with some disease symptoms, so actually we can tell if you have that antibody, and then by telling that, we're gonna try to discover if you have that disease or not. Actually, we're looking at patterns since we have 2,500 spots, so we're gonna show you how they look like.
A system that we study called microarrays. If you look in here, we have a synthetic microarray. It's a light bright. On this side is one pattern, and this is healthy. Can you see that one? Yeah. Then you look over on the way right, your way right. That person has a sick pattern, and now we have somebody in the very middle. And you have to decide whether they're healthy or they're sick. Can you help us? Can you help us treat this patient? Or does it need treatment? Does this patient even need treatment? You think it's a healthy? Excellent. All right. That person doesn't have to go to the doctor until next year.
Good job.
Okay, I'm gonna give you a. We're gonna switch it up.
Okay.
You're not gonna know who's what. Okay. Now take a look. So over here, way on the right, on your left, sorry, is healthy. Way on your left, on your right, is the sick. Look at the arms and the
expressed. So hit 2 because that's indicating. Now I also see here there's some individual proteins which look cancerous. All right, you got 4 on that, but start over and just see for 30 seconds how many you can properly assess whether you have cancerous or healthy tissue. I see you're going really fast there. There, you see, you've looked at microarrays before.
I'm just looking for red.
Yeah, well, that's what we're looking for. We're looking for the highly expressed proteins, which are indicating, some kind of tissue which is out of whack. Now, when you got 28, that's a very good score. 28, all right.
Thank you.
But when we actually do it, of course, our lab will use like about 2,000 spots on a slide, and
Make you dizzy to watch all those spots?
well, I don't look at spots. We actually have scientists look at spots. They'll use software, and they'll look at. They're not trying to look at 130 seconds. They're going to look at one slide. One slide might cost $100 or $500 to generate, and they're going to look at that and they're going to analyze that. They'll use software. They'll look at it under a microscope or on the screen, and we know every one of these spots is a specific protein. Now we might have maybe 5 specific proteins that I've seen again and again. That indicates a certain cancer, and so I'm going to just look for those spots. But what if I don't know which spots? Then what I'm trying to do is compare. Maybe 100 patients have this cancer, and I'm going to look at 100 scans, 100 slides, and each of those slides has 8,000 spots. And I'm trying to compare and see which of these are cancerous versus which are not cancerous. What's the pattern? Right now we're just trying to figure it out, It's there, the information's there, but it's hard for us to figure out who has cancer versus who doesn't have cancer just by looking at these spots. There's variation in people, so it's hard for us to just isolate. Now, companies have spent millions and millions of dollars just looking for those 5 spots which indicate cancer. And if they can find those that are always indicating cancer, they got a marker. They got something they can package. They can give to doctors. Doctors can say, I can do this test in a few minutes, and I know that you're going to be sick in 6 months. Right now, we don't know which 5 spots those are. And there might not be 5 spots for everybody. It might be some people have these 100 spots, some people have these 50 spots. That's the thing. We have so much variation in the gene pool and in the population and in your environment. The same test isn't going to work for me that works on you. Yeah, that's why we have personalized diagnostics. So we're trying to figure out the code. Thanks.
Does that travel? Does that go on the road? Can I take that with me?
I just made it this afternoon. I can give it to anybody. Yeah. Looks like you've got the hang of it. You played this before. You were here before, yeah.
came to the dimension of head of the pin or like a dust mite. But if you want to go to the nano dimension, you have to cut it 24 or 23 times. Now, can you imagine how incredibly small it is, and it is impossible to
see?
Huh?
It would be impossible to see.
Yeah, but we have advanced machines and things to see them. And now Palash will give you an idea of when we go from a macroscopic world to a nano world, the property and the rules changes totally. Okay, now he will give you an idea how the properties changes when you go from a macroscopic world to a nano world.
Hi, welcome to the nano world. We have seen the gold, right? This is light yellowish color, and the silver ring, which is metallic color or light gray. Now, if you break this gold ring to tiny pieces like 50 nanometer, so you know 50 nanometer? Smaller than this, so you need to cut this more than 21 times. So how small it is, right? If you cut it to 50 nanometer, you get this. So yellow thing, this is yellow. And if you get it 50 nanometer, you get red color. For silver, it gets yellow color. So in the nano world, the rule is completely different from what we see. How they behave, how they look, everything is different. And this is our research. We want to understand how they behave. I think you use sunscreen lotion, right? In the sunscreen lotion, they have nanoparticles, and that particle can absorb UV light. So if you know how nanoparticles behave, then you can make use of that. And that's why we are doing this science. More specifically, we also work with DNA structures. Like, you see the bone, those structures, all those with DNA. We have DNA in our body, right? So using DNA, you can make the structures and you can study them, how they behave, how they look. Now, one question may arise, in your mind. How you can make these tiny particles, right? How? Just any idea? No idea. Okay, so there are two ways. One is you have a big rock. If you break it down, you can make a small sculpture, right? Similarly, if you have a big gold, and if you break it down to small, tiny pieces using some hi-fi, ultra-modern instruments, you can get micron size or nanometer level. This is one way. Another way is this one. Have you played this before? Yeah, sure. So, if you join the small blocks, you can get this Mario, right? Similarly, do you know what atom is? Atom? So if you join small atoms, you can get these structures, 50 nanometer or few hundred nanometer structures. So there are two ways to make these structures, and there are a lot of other ways to visualize these structures. You can see this in naked eye. You can see the color, but you can actually see the structure, which is 50 nanometer, using some ultra modern instruments. Phone call.
And we're producing a compound which can be used for powering jets or cars or anything like that.
So the inputs are carbon dioxide and what else? You've got bacteria?
Oh yeah, so the bacteria are basically the reactors, right?
Okay.
And they just use light and carbon dioxide to produce the fuel.
Oh, okay, so what you're doing here is free.
Yeah, it's
for all practical purposes.
For all intents and purposes, it's free to produce.
Okay.
The fuel's not green. No, it's the bacteria that are green, right? So they're when the light hits them, basically the only color that they don't absorb is green. So that's why you see the green. Yeah.
This is the membrane biofuel reactor, and we grow bacteria here to clean up water. So the bacteria are capable to transform contaminants into harmless substances.
Put this in the water and let go. So where do you find the bacteria? Get rid of the water.
These are strains that have just been around, cultured in the lab for a really long time.
Okay.
This is just a wild type strain. We're using this particular one because it was the first to have its full genome sequence, which it's really well known, really well understood, and it makes it easy to genetically modify it.
Okay.
And so this strain has been genetically modified, and these are the fatty acid producers. And if the culture was a little bit older and a little bit thicker, you could actually see there'd be a white strip around here, and you could see that that's the fatty acid.
Okay, that sounds pretty cool.
Yeah, it's cool. It's a good technology. It's fun. I'm working on scaling it up to a commercial process, basically.
Yeah, I heard the defense department's starting to buy fuel and Virgin, actually flew a jet. I forget where they started flying from. They did fly a jet, I think, across the Atlantic Ocean.
Oh, really?
Yeah. Did about a year, maybe 2 years ago. It's pretty cool. I heard about it. It's biofuels.
Yeah, everyone's really getting into the biofuel thing.
Yeah, definitely.
Yeah, we're no exception to that.
Will grow on different sugars, and so these different agars have different sugar, and based on the colors they turn, you can tell the different types of bacteria. So if you have a mixture, you can isolate them easier. And there's a rose.
Hey, that's cool.
That's a shamrock.
Shamrock.
This is the owl, right? That's on all of our T-shirts.
Oh, that's awesome.
Right, so those are two different bacteria. And here you can see that Salmonella grows black and E. coli grows yellow on this plate, which is XLD, which it originally starts out as a red. Would you like to try some pipetting?
You want a pipette?
Okay.
We would monitor the air quality through a system that will see the quality of the air that's in there, and when the quality of the air is really good, it reduces the air volume in the space down. And so if somebody takes a lid off of an alcohol bottle, the system will sample that, and it'll start to ratchet up the air volume to dilute it, so the customer doesn't even know that there's excess amount of total VOCs in the air. And then when they recap it, then the system will ratchet back down. So that gives us a big savings, especially at night when there's nobody in the labs. Let's go ahead and ratchet down as much as we can, and that's where we're seeing some of the savings. That's just one feature of the building.
Hey, buddy. Here you go.
Thank you. It'll be about 5 minutes if you want to do it. Are you guys having a good time?
Is that better?
Sure.
What would you like me to say before you record?
Please just tell me about your booth. What you got going on here? What exactly? What's going? What is your booth and everything like that?
So to think about that. What is our booth? Okay. Hi, my name is Annie Hale, and I work for the Center for Sustainable Health. At the Center for Sustainable Health and at our booth, we're looking at two different things. One is sustainable aging, which is very interesting, and what it means to live in a sustainable area, what it means to have mobility and so forth, and aging in place, and these sorts of questions are being tackled in our center. And another interesting thing is how to teach future teachers, K-8 teachers, about sustainability and science. So we create a larger electorate of future teachers that will go out and tackle these tough questions of water pollution, food, and so forth. So here in our booth, we're actually asking people where they think the driest place in the world is and where they think the wettest place in the world is. We're also talking about food miles. Did you know that 80% of all canned pineapple comes from Thailand? It's a very interesting little fact. And Chiquita bananas actually travel roughly 3,000 miles from farm to fork. So these are some of the facts that we're talking about here at our booth, and sort of lead into the larger questions that we talk about at the Center for Sustainable Health.
Alrighty, well, thank you very much.
Thank you. Have fun. Come back.
Courtney.
How you doing? Oh, hey.